Welcome to the second episode of my molecular biochemistry series, and in this video we are elaborating on the structure of DNA. Now, I did talk about the strand of DNA and its molecular makeup and how we can remember some of the nucleotides, the 5' 3' anti-parallel nature of the DNA strand, but it's important to note that the DNA cannot operate in isolation. It does not sit in the cell by itself doing its thing. It is actually a bit more elaborate than that. It actually coils around a protein known as a histone. The histone is a protein that has a positive charge due to lysine and arginine amino acids, which are basic amino acids that give off a positive charge. And that is how DNA is bonded to the histone, given that DNA has phosphate groups that give it its negative charge. And so it coils around or wraps around the histone multiple times, and when it does this, it forms what we call a nucleosome. Now, this happens over and over and over again and is connected through something called linkers, and it does this until you get a super coiled structure that makes up what we now call the chromosome. Now, all of this sits in the nucleus of the cell typically, and it can be modified in several ways. We can add an acetyl group to interrupt this negative-positive interaction, and it can uncoil the DNA from the histone. And this actually is an epigenetic approach to increasing transcription and forming what we call euchromatin. Alternatively, we could remove acetyl groups, and this would allow the DNA to once again loop around the histone, and we call this heterochromatin, and this actually decreases transcription. And interestingly, oftentimes you would hear people talk about, researchers, scientists talk about how DNA is incredibly long, and there is so much of it in our bodies, within our cells, that one DNA strand can be up to 6 feet long in a given cell, and then we have trillions of cells, so you can expand that to be 60 trillion feet. And this is roughly 120 billion miles of DNA sitting inside us, which is impossible to imagine, but that is twice the size of the solar system. This is all interesting, and there's a whole entire category or area studying this in biological topology, which is something that is extremely fascinating and is relevant to protein folding as well. But topology as a mathematical area is also very interesting, that I know very little about, but it is nonetheless so cool that you can just wrap around a ball, a piece of rope, and you can fit so much of this structure called DNA into such a small area. And maybe we can apply this idea in other domains and disciplines in human life and take advantage of this concept. But anyways, I continue to rattle on. I'll try to move on here. And the other important thing I wanted to note is that we can actually add and take away methyl groups in addition to acetyl groups. And if you remove a methyl group, it actually will promote gene transcription. But if you add methyl groups, it represses gene transcription, similar to removing a acetyl group. Adding a methyl group mutes the DNA. And one interesting or notable syndrome that develops from this is called Fragile X syndrome. And this dysregulation is driven by the fact that a sequence of CGG is supposed to repeat roughly 40 times, but in the unfortunate scenario where you have Fragile X syndrome, it repeats almost 200 or 200 plus times. And this is constantly hypermuting the promoter known as, or the gene known as FMR1, and you get a various different Symptoms due to this methyl group consistently muting this gene, including intellectual and developmental disabilities and autism spectrum disorder. So this is just one area where this is relevant in human life, and it's actually more aggressive or severe in males than females, even though it sits on the X chromosome. So this is just one more topic that we are studying in our molecular biochemistry series. We have looked at the structure of DNA quite closely at this point, and we will further look into what happens when we mutate, and actually we'll look at nucleotide synthesis first and look at how we actually get the nucleotides that sit in a DNA, because they don't just appear out of nowhere. Our bodies need to synthesize them in a thermodynamically favorable manner. And we'll see you in the next video.